Bonjour, good morning. My name is Eric Aliwa and I'm the president of CEO of PDC Line Pharma, and I'm going to present you over the next 20 minutes the activity of PDC Line, which is developing a new class of cancer vaccine based on an off-the-shelf antigen-presenting cell line. In a nutshell, the company is a spin-off from the French blood bank. In fact, the scientific founder of the company has identified, when he was leading an academic research lab associated to the French blood bank, a very rare leukemia affecting the plasmacytoid dendritic cells circulating in the blood of the patients. And this leukemia is extremely rare because in the dendritic, you have a majority of the cell are the myeloid, and the plasmacytoid is a very small portions of the cells. And based on the blood of these plasmacytoid dendritic leukemia patients, he developed a master cell bank, GMP approved and for clinical use, that we are still using today as a vaccine platform. We're a team of 25 people, including seasoned executives from biotech and pharma industries. In terms of funding, we get close to 35 million euros of funding so far. We will announce soon a new round of financing at the top of this 35 million euros. Our proprietary PDC line have three key advantages. First, it's an immortalized cell line. It's a leukemia cell, so it's very easy to expand. And I will show you the manufacturing process later. Secondly, the origin of the cells. These plasmacytoid dendritic cells have very interesting biological property and capacity in terms of priming and expanding CD8 cytotoxic T cells. And last but not least, the cell, the platform is very versatile. In fact, we just have to stick to the HLA present in the membrane of the cells, GMP peptide representative of the tumor antigens. And in this way, we can potentially address any kind of solid or liquid tumors. We are a clinical stage company. We have already completed a phase 1b academic study in melanoma on 9 patients, and we have an ongoing study in lung cancer, a phase 1/2 in non-small cell lung cancer. The pipeline of the company is briefly presented here. We have 3 components: the PDC-MEL, the melanoma vaccine, which is not active anymore. The result of the clinical trial has been published last year. The second component is PDC-Lung, the cancer vaccine candidate for the non-small cell lung cancer with the 6 shared tumor antigens, which is ongoing in phase 1/2. And we have the neoantigens-based vaccine based on specific pool of frameshift-based mutations, peptide-related, that we are working on currently at preclinical stage. Important to mention that we have already signed a first licensing deal with LG Chem in Asia for the lung cancer vaccine. We provided exclusive license in South Korea and options for the rest of Asia. And you see on the bottom of the slide the total deal value, which is over $123 million. So we have a first partner in Asia, but we are open to new partnerships for other indications or for new indications we could explore together. The management team of the company is one of the key assets of the company. Of course, at the top of the technology, we are very experienced people. We have the medical director, Shanna De Bruyne, has a long experience in cancer vaccine. She comes from GSK Vaccine, where she led the clinical trials, the late-stage clinical trials in lung and melanoma cancer vaccine of the MAGE clinical trial. Claude Edry, our CMC director, has a long experience as well in cell therapy and is a former GSK Vaccine. The scientific founder, Joël Pluma, is the co-inventor of the technology, joined full-time the company. Myself, I've co-founded already several biotech in the past. I've been involved with several exits like IPO or M&A. I sold one of my company to Genzyme. And Laurent Lévy, the CFO, as well, has a good experience in oncology. In the board of directors, we have as well very competent people like Monder Majoubi, who is the former senior executive VP of Genentech Oncology, AstraZeneca Oncology, and current CEO of Innate Pharma. He's the president of the board. Jean-Paul Prils is the former R&D director of GSK Vaccine. And we have representative as well, of course, of the investor, like Korea Investment Partner, which is one of the leading South Korean investor and a leading investor in Asia, private equity in Asia. The company made significant achievement over the last 24 months. We have been accredited for the GMP accreditations by the Belgium Federal Agency for QC and release activity of the PDC lung vaccine candidate. We closed, as I said earlier, the deal with Algichem in March 2019. We get the authorization to launch a phase 1/2 trials with PDC lung in non-small cell lung cancer in France and Belgium. In June, in December, we closed a 20 million B round of financing led by the Korean investment partner. In February, we announced the administration of first patients with the low dose of the vaccine PDC lung 01 for the non-small cell lung cancer. We are testing 2 doses. In April, we published as well of international patent application on a modified PDC line secreting a specific cytokine. The president of the board, Mondher Mahjoubi, joined the company in June 2020. In September, we announced the administration of the first patient with the high dose. In November, we get authorization to launch the phase 1/2 trial in Germany. And in December, we announced as well the 100% compliance of all the batches of the PDC lung vaccine candidate that were manufacturing. So showing that the CMC is completely under control. Okay, let me explain you now how the technology works. As I said earlier, we have a master cell bank where the blood from the PBMC of the PDC leukemia patients, we have purified the cells, the specific leukemia cells, put in bag, and these bags are in liquid nitrogens, and they constitute the master cell bank. From the master cell bank, we have developed several working cell banks, and from a bag of a working cell bank, we just have to seed a single-use bioreactor. We have today 1 liter and 10 liters bioreactor. The cells can grow in suspensions in 9 to 10 days, and we can have more than 20 billions of cells. Then at the harvest, the cells are loaded individually by each peptide according to the number of peptides we want to load in bags. So we load with the GMP peptide loaded in the bags, protein present in the bag that will stick to the HLA present membrane of the cells. Then we pull all the bags and the cells. We irradiate the cell to stop the proliferation. So I remind you, it's a leukemia cells. Then we put a specific container, the technology of aseptic technology with a cryoprotectant. When the patient is ready, we just have to, like for the COVID-19 vaccine, to ship the cells in liquid nitrogens. And when the patient is ready, in minus 80 degrees fridge, sorry. And when the patient is ready, we just have to thaw the cells and with a syringe, take the cells from the vial and infuse to the patients. And the vaccine will make the job, meaning to prime, to go to the lymph node, to prime naive and expand memory T cells that will migrate to the tumor microenvironment to kill the tumor. We did several benchmark studies to demonstrate the capacity, the potency of our cell line to expand cytotoxic CD8 T cells. And you see here one example of a benchmark study we have published where we have loaded the cell line with Melanoma, which is a specific antigens, and loaded as well conventional myeloid autologous and allogenic dendritic cells, and measure by fast dextramer the expansions of CD8 T cells after specific of the Melanoma after 21 days of culture. And as you can see here in this graph, we are 20 to 200 for more potent than the other approach. We did as well study with neoantigens, and for that we have selected 2 mutated peptides identified in the blood of melanoma and lung cancer patients, where the mutations you see in red in amino acid has been clearly identified and characterized, and we have loaded our cell line with each of these antigens and measure by FACS dextramer the expansions of the CD8 T cells specific of this neoepitope in the blood of 3 healthy donors. And as you can see here in red and purple, the expansion by FACS, D0, D7, D14s, D21s, the D21s, we are able to increase by more than 100-fold increase the quantity of neoepitope-based CD8 T cells from the blood of this healthy donor. And that is extremely interesting because, in healthy donor, all the precursors are naive, and the prevalence of this naive CD8 T cell is extremely low, is 1 out of 1 million. So showing once again the potency of our cell line. Okay, so today we are presented HLA-A2 restricted peptide because the patients was the Caucasian patients and the HLA-A2 is very prevalent, but there is minority HLA expressed by the cell line like the B7 or B44, but we have modified genetically the cell line to express additional HLA like the A24 and B35, and we have published recently this result. We can definitely use A24 B35 restricted peptide and expand very effectively the cytotoxic CD8 T cell specific of each of these peptides. In terms of loading, antigen loading strategy, today we are using GMP peptide that load on the membrane of the HLA present membrane of the cells, but we have already done over experiment with retroviral transductions to have endogenous expression and presentation through the HLA of the polypeptide, and it works quite well. We did as well electroporation of mRNA. It works very well as well. So we have a very versatile technology, and we can adapt to the loading strategy according to the indications. In terms of IP strategy, we have three key pillars. The first one is the master cell bank, which is unique. In fact, the patient is dead now, and we have a clinical grade master cell bank approved by the French authorities, and we have exclusive license from the French blood bank. Second pillars is the IP, the patent, sorry. We have 3 patents. 2 has been granted related to protecting the dendritic cell line or the applications of the dendritic cell line in active or adoptive cell therapy. And the third one filed in 2018 on the genetic optimization of the cell line. And last pillars, the third one is all the know-how and data related to the manufacturing process, QC, immunomonitoring assay, and preclinical data. Okay, let me show you now quickly the result of the melanoma clinical trials. So we have done a phase 1B academic study on 9 patients with advanced melanoma, which was a phase 4 patients after 3 or 4 lines of treatment, where the vaccine was infused alone, and the vaccine was constituted of the cell line loaded with 4 different antigens that you see here: tyrosinase, GP100, melanase, and MAGE3. The results were definitely promising. We have observed acceptable tolerability and safety profile. That was the primary objective. We didn't observe any allogenic rejection detected, and we have observed as well significant antigen-specific CD8 T cell response that were directly detectable in the blood of the patient. So meaning without any in vitro restimulation. So showing that the vaccine was biologically active in human. And that was interesting because we just infused 3 times weekly the vaccine, and we didn't administer any checkpoint inhibitor co-administered with the vaccine. And we know that there is synergetic effect between the vaccine and the checkpoint. And you see here an example of the result that has been published. You see an example. We have, to demonstrate the synergetic effect, we have taken the blood of melanoma patients. Melanoma is a very hard tumor with a very high tumor-induced immunosuppressions. And we measure after 1 week of culture, the capacity of the cell line to expand melanase CD8 cytotoxic T cells by fast dextromer. And as you can see, after 1 week with the vaccine alone, we have 15-fold increase. With the checkpoint inhibitor, we have 2-fold increase from baseline, but it's not statistically significant. But when we combine both the checkpoint inhibitor and the vaccine is where you see this very nice synergistic effect with a 35-fold increase. This data has been once again published in Oncoimmunology in 2020 and available in our website if you're interested to read it. Okay, let's pass now to the non-small cell lung cancer trials. You see here the 6 antigens we have selected: MAGE4, MAGE3, MUC1, New York ESO1, Zurvivin, and Multimage. And for each of these antigens, with the blood of healthy donor, we have been able to demonstrate a strong expansions of CD8 T cell cytotoxic specific of each of these antigens. It's after 2 weeks of culture, and it works better for some than the others, but for all of them, we have a statistically significant increase and or expansions of CD8 T cells, and that has been demonstrated for each of these patients. You see here the design of the current clinical trials. It's on 64 patients. It's ongoing now in Europe, in France, Belgium, Germany, Netherlands, and soon in Poland. We want to enroll 64 patients. Main objective is to assess safety, immunological activity, and clinical response of the vaccine with and without checkpoint inhibitor. We will test 2 doses of vaccine, the low dose and the high dose. A low dose monotherapy first in adjuvant setting for patients phase 3B for phase 3A, where the surgery is feasible. And so you have 6 patients in monotherapy, then 10 patients in monotherapy with the high dose. You see here the 2 different doses, 2 million cells per peptide for the low dose and 2 million cells per peptide for the high dose. The vaccine is infused 6 times and not 3 through IV and subcut weekly. And after the 2 last cohort is in metastatic patients, first line for PDC checkpoint inhibitor naive patients, and the vaccine is infused in combo with checkpoint inhibitor. First with the low dose, 6 patients of the vaccine, then we go to the high dose with 42 patients. And so this is the design. So we have already completed the 3 first cohort, opened the last cohort, and infused already the first patient. So it's still ongoing. To finalize my presentations, you see here the timeline and the funding strategy. We have already closed the 20 million B round of funding. We expect to raise an additional close to 80 million round of financing before the end of the year to complete the current clinical trials, the 64 patients. We expect to have the clinical study report end of 2023. And at this time, after this report, we want to make whatever an IPO, a big licensing deal with a big pharma, or to finance a new clinical trials in partnership or alone, and to set up in US, in Asia, or to launch new clinical indications. And I would like to thank you for your attention, and do not hesitate to contact me by email if you have any questions related to my presentation.